Okay, let's talk about classical genetics. First, we have to know something about the law of independent assortment. So basically, in this law, it's stated that each member of a pair of homologous chromosomes, they basically separate independently of other pairs, and the result would be is that they'd be random. And yeah, as chromosomes segregate, they are not influenced by genes. This is important to know. So yeah, genes are not unimportant. Yeah, they're not important. They're not they don't influence chromosomal segregation, and genes are independent from each other. So for instance, we have albinism. For albinism, there are genes on hair color of a mouse and another for whether mouse produces pigment or not. So um, basically in this case, let's say there are certain mice that are color brown and there are certain other mice that are color excuse me, they're color gray. And there, even though the parents, the mother or the father, are either of those two colors, let's say we have a brown mouse that's a female and a gray mouse that's a male, then there's a chance that the offspring would be albino. That means they're white. They wouldn't have any pigment. So yeah. Something about Gregor Mendel. He, by the way, is the father of classical genetics. Why did he choose garden pea in his experiment? So first things first, garden pea grows quickly, and secondly, they're easy to breed, and thirdly, they have a variety of traits. So he thought that garden pea was the best kind of plant that could be used in order to study more about this uh, scientific discipline, um, which was very new back in his time. And so, yeah, how Mendel's bees help understand genetics. So, principles that rule heredity. First, each trait depends on allele pairs. So, allele basically represents different variations of a gene. They're different from they're a different form of gene at a loci. A loci is a specific location within a genome. A genome is basically a gene. Please correct me if I'm wrong in that part. So homozygous, both alleles are identical. They're pure breed. Uh, Heterozygous, two alleles are different. For genotype, this is the combination of alleles, and phenotype is whether it's trait A or trait B. So it's basically the physical manifestation of traits. For genotype, it's just basically the symbol that describes the phenotype. Uh, that's I worded it in my own way, but I know there's a more formal definition to that. For example, the genotype is here based on this Punnet square. We have um, for the offspring we have for basically 100% of them have this symbol. And what does this mean? Heterozygous yellow. So for the phenotype, it's basically the translation of what this symbol is. So yeah. So what is the Punnett square in the first place? It's basically intended to visualize how alleles are distributed among descendants. So coming from the mother and the father. Okay. So usually in a Punnett square, uh, we use these symbols. This one is for the male. This one is for the female. So this one is, for instance, this part will be for the male and this part will be for the female. But in this case, it's not really that important, especially when we're dealing with plants. I'm not sure. Maybe that's something that we could talk about as a basis for discussion. But surely, we use those symbols in Punnett squares that involve humans and animals, definitely. But for now, I think this is about flowers because, yeah, We're talking about the flower's color. So let's focus on this one. So if we breed two um, two species that are both homozygous yellow, then in the Punnett square, all of the offspring, 100% of them, will be the same kind. So they'll be homozygous yellow. And for the phenotype, it's the same thing, but basically the translation of what this symbol is, the capital G. Let's go to this one. So if we breed this time, both of them are homozygous, but one has the dominant trait because the letter is capital, and the other has the recessive trait because it is not in capital letters. But even so, I suppose the recessive gene, the one in small letter, stands for color green, homozygous green basically. And if we cross them, so the outcome would be all of the offsprings will be heterozygous. They'll be getting the capital G from this species and the small g from this species, and then they'll be getting they won't be homozygous, they would be heterozygous. And the color, the capital G stands for yellow, right? So whatever is dominant will be the dominant color. So instead of writing green, we write yellow. So 100% capital G small g, or in other words, heterozygous yellow. So if you can see the difference between this and this, if it's both capital or both small, then it's supposed to be homozygous. If there is a capital and a small letter at the same time, then it's heterozygous. Okay, moving on. We have a heterozygous capital G small g, and then we have small g. Heterozygous crossed with a homozygous. So what would be the outcome? So plus g small g, plus g small g, small g small g, small g small g. So for these two, they since we have 4 boxes, 100% is equivalent to 4 boxes, and 2 of them are heterozygous yellow. So capital G small g is equal to 50%, and small g is equal to 50%. And yeah, what I said earlier was correct. The small g is for green, and since it's both small letters green, both small letters G, then it's homozygous, and yeah, it's green. What is the phenotype? So we have these two for the genotype. Capital G small g is heterozygous yellow, and small g is homozygous yellow. And we write the percentages again. Okay, what's the other one? So this time we'll be crossing 2 heterozygous yellow species. So what would be the outcome? Generally, so we have 2 capital G's here, 1 heterozygous here, another heterozygous there, and a homozygous green here. So 75% is yellow. 75% is yellow and 25 is green. But in terms of if you try to segregate them according to heterozygous and homozygous, then definitely this is the 25% is homozygous yellow, 50% is heterozygous yellow, and 25% is homozygous green. So yes, this is more specific compared to when you just say that 75% of the offsprings would be yellow and 25 would be green. So I'm not sure which is more relevant, but this is what is written anyways in the reviewer. So yes, I should have written this here. You should have known this earlier. I should have shown this to you. So capital G stands for yellow, small g stands for green. Okay. So there is certain, I'm not sure what these are formally called, but we have something called dominant and recessive, incomplete dominance, codominance, and sex-linked. More on, actually, yeah. Also, we have in a dihybrid cross, we have dominant, recessive, or sex-linked. Okay, so let's focus on these two first. So what's an example for dominant and recessive? So we have two heterozygous species. I suppose, yeah, it's stated here that capital T is tall, small t is short. So if we cross them together, we have capital T, this is tall, capital T, small t, this is still tall technically, and small t is short. So what is the genotypic ratio? This is basically another fancy word of saying genotype, but with a ratio. So we have, for homozygous tall, we have 25%. Heterozygous tall is 50%, and homozygous short is 25%. So if you put it in a ratio, uh, basically the total sum is supposed to be 4. All of the numbers in the ratio should amount to 4. So this is 25%, this is 50%, and this is 25%. So 4 is basically 100%. I suppose that's basic logic. Let's go to the phenotypic ratio. So for the phenotypic ratio, we have 3:1. Tall is 75%, definitely. Okay. Um, I think I understand it now better. So for the genotypic ratio, we need to consider the homozygous and heterozygous. But for the phenotypic ratio, we only want to know how much is this trait and how much has the other trait, which is the recessive. So for tall, 75%, short, 25%. So 3:1. So that's the ratio for the phenotypic aspect. Incomplete dominance. So provided that we have, yeah, technically we have the same thing. Okay. The only difference between this Punnett square and the one we did earlier is that instead of being heterozygous, I suppose it's technically still heterozygous. However, if there is incomplete dominance, then this is supposed to be medium. Instead of being either tall or short, usually you become tall, but the tendency is the dominant trait becomes incomplete. So if you're heterozygous, then your height usually becomes medium, something in between tall and short. So yeah, what is the genotypic ratio? The same thing. Phenotypic ratio, well, instead of being 3:1, since we have another trait called medium as a result of incomplete dominance, uh, we only have 25% tall, 50% medium, and 25% short. Codominance. This is the third one. Provided that we cross, I think the example here is basically chickens. So we have a black chicken and a white chicken, and if the heterozygous variant provided these two homozygous variants, we have a tan chicken, or I think this is chicken that has traits coming from both black and white. Since both traits, black and white, are dominant, there's a tendency for codominance, hence the word codominance. Okay, so if we cross BW, we have here we have black, 2 tans, and 1 white. So genotypic ratio 1:2:1, and the percentages over there. And phenotypic ratio the same, but this time we just label the symbols based on what they actually mean. So BB is black, BW is tan, WW is white. Okay, how about the sex linked? So if we cross, I think this is regarding males and females this time. There you go. So this is for female. I'm not sure what this is formally called, but female is associated with 2 variables called XX, and for males we have XY. So if we cross XX, wait, before I start with saying this, let's start with this first. So H is equal to normal, and small h is hemophilia. Hemophilia, by the way, is I suppose an X-linked recessive trait. So yeah, that's all I'm gonna say. So if you cross a female with hemophilia, I'm not sure if this is a female with hemophilia or perhaps just a carrier. Yeah, definitely a carrier, because if someone has hemophilia for females, it has to be XHXH, but that's not the case. I also want to tell this to you in advance. If it's a male, let's say, yeah, we have whatever this is. Males cannot be carriers. It's either they are unaffected by the disease or they are affected by the disease. So they can't be carriers. Maybe I suppose it has something to do with the fact that they are XY, they're so Y. And for females, they can be carriers because they're XX. It has something to do with their X variable, because again, hemophilia is an X-linked recessive disorder. So, okay, if you place these in the Punnett square, X, X, and then we have H, X, H, and then Y. So XXH, again, this is a female carrier. XY, this is normal. Regardless if you don't have the H, the capital H, if there is no small h, then definitely it's gonna be normal. It means normal. And we have X small h, and then X small hy. This one represents hemophilia because, again, as I said earlier, in order for a female to have hemophilia, both X variables should have an H exponent, basically. In terms of algebra language, that's how you say it. And here, X, H, and then Y. This is, the male has hemophilia. Okay, yeah, that makes sense, I suppose. Yeah, the male has hemophilia if the letter has a small h. If the X variable has a small h. Because again, hemophilia is an X-linked recessive disorder. X-linked meaning it somehow binds to the X variable of regardless of your sex, if you're male or female. Everyone has an X variable. I know it's not right to say the word variable. It has another word for that. Maybe gene, X gene. I'm just not sure at the moment. Okay, genotypic ratio. So based on the Punnett square, everything's different. So we have 4 different types. This one. Okay. Another concern is that I don't think I've asked my teacher this question, but is this the same as this? Technically, I mean, yeah, this is a question that's been unanswered, but I guess we'll just have to deal with it ourselves. So this basically is for the female carrier. This one is for the female with hemophilia. This one is the normal male. This one is the male with hemophilia. So yeah, phenotypic ratio basically describing what the symbols mean. Okay, let's move on to dihybrid cross, dominant and recessive. Let's check where we could possibly end our discussion. Sorry about that. Okay, this is also one of them. We have something called multiple allelism. I'm not sure if it's this one, but yeah, this table is important to memorize. So we have blood type, the phenotype, and the genotype. So if we have the phenotype of type A, then the genotype it corresponds to the genotype of AA and AO. For type B, BB and BO. For type AB, we have AB. For type O, it's basically OO. I'm not sure if I'm saying that correctly. Okay, so for example, let's cross two species. One has AB and the other has AO. So we have AA, AB, AO, BO. Genotypic ratio, for AA it's 25%, meaning this offspring has type A, blood type A. For AB, this offspring has blood type AB, basically. For AO, similar to the first offspring, he or she has type A, and BO corresponds to having a blood type B. So 25% for each. They're all different from each other, four of them. And yeah, phenotypic ratio, just translate the symbols to what they mean. And yeah, this is also another question that I asked to my teacher some time ago. I think the exam was taken roughly more than a month ago. Is it not 2:1:1? Because technically, type A is 50%, right? But even if this is already written, the ratio is still 1:1:1. So there must be something wrong. I got this worksheet from the internet, by the way. So yeah, for the purpose of being a teaching material. So before we go to this, let's try to, never mind, I already wrote them anyways. Yeah, this is my ratio instead of this. Okay, let's go here. So dihybrid cross is basically a lot bigger compared to what we've been dealing with in the past minutes. So in the past minutes, we dealt with basically a monohybrid cross. But right now, it's a dihybrid cross. It's a bigger Punnet square. And instead of having normally two traits, we have four. So capital T is tall, small t is short, capital B is black, and small b is white. So granted that we have to cross capital T, small t, capital B, small b with the same thing. So we write them down in a Punnet square, and yeah, we write the offspring. So capital T, capital B, capital B, capital B. Capital T, capital T, capital B, small b. And we just keep on going. And then from there, again, if it's a capital letter, then that's the trait that you'll be writing. And if there's a small letter, if there is no capital letter, then this is what you write. So here, capital T is tall, capital B is black, capital T is tall, capital B is black. Just keep going. Here we have capital T is tall, and then small b is white. So those are highlighted purple. 3 of them matched together. The gray ones are the same phenotype and not necessarily the same genotype, but they have the same phenotype. And for the green ones, we have small t, that's short, and capital B, which is black. And then 1 matches for both short and white at the same time. So what's the genotypic ratio? Well, it's already stated here, but yeah. It's stated that the genotypic ratio, capital T, capital B, capital B, capital B, we have only 1. Although everything from the gray highlight are of the same phenotype, the genotype is still different from each other. So we just diversify them basically. So 1, 2, 2, 4, and then 1, and then 2, 1, 2, and then 1. And then for the phenotypic ratio, it's a lot easier. It's 9:3:3:1. 9 coming from the gray highlights, it's 9 of them. 3 purple highlights, 3 green highlights, and 1 light blue highlight. So that's for the phenotypic ratio. Usually, what's being asked in an exam is only the phenotypic ratio if you're dealing with the dihybrid cross. But granted that you have to look for the genotypic ratio, good luck with you. It's gonna be more challenging, but just make sure that you're focused, because it's easy to make a mistake in this part. And here, it's basically the same thing. So yeah, we'll be talking more on that later. See you.